This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome. Have you ever wondered why we have lips? Today, we'll explore the fascinating journey of how lips evolved over millions of years, from simple mouth openings in ancient fish to the expressive, sensitive features we have today. We'll discover how lips helped our ancestors survive, communicate, and eventually speak. Along the way, you'll learn about the incredible adaptations that transformed these structures from basic feeding tools into one of the most distinctive and functional parts of the human face. Let's dive into this remarkable evolutionary story. Chapter 1. The Curved Muscle Ring. When you look in the mirror and notice your lips, you're observing one of the most distinctive anatomical features that separates humans from every other creature on Earth. These soft, pinkish structures framing your mouth represent a remarkable configuration, one that goes far deeper than their visible surface. Beneath the skin lies a muscular architecture so uniquely designed that it enables capabilities no other primate possesses. At the heart of this anatomical marvel lies the orbicularis oris muscle, a ring of muscular tissue encircling the mouth opening. While all primates have this muscle, the human version has evolved a configuration found nowhere else in the natural world. In humans, the orbicularis oris forms a distinctly curved, hook-shaped structure. This may sound like a minor detail, but it represents a fundamental redesign of the oral apparatus. In chimpanzees, gorillas, orangutans, and all other primates, this same muscle maintains a flat, planar configuration. The difference is not merely structural, it is profoundly functional. To understand why this curved configuration matters, we need to examine how the muscle operates. The orbicularis oris functions as a sphincter, a circular muscle that can contract to close an opening. Think of it like a drawstring purse, tightening to seal the mouth shut. However, the human version does far more than simply open and close. The curved geometry of our orbicularis oris allows for an extraordinary range of precise movements. We can purse our lips, pucker them, stretch them laterally, protrude them forward, or retract them inward. We can perform these movements with millimeter precision, adjusting the shape and position of our lips independently at different points around the mouth opening. This level of control is unprecedented in the animal kingdom. When a chimpanzee moves its lips, the flat configuration of its orbicularis oris limits the range and precision of motion. The muscle can contract or relax, but the planar structure restricts complex three-dimensional shaping. Human lips, by contrast, can assume an almost infinite variety of configurations. We use this capability constantly, though we rarely think about it. Every time you pronounce the letter P, you are bringing your lips together with precise timing and pressure. When you say the letter F, you position your lower lip against your upper teeth with exact placements. When you round your lips to say the word who, you are creating a specific aperture size that filters sound in a particular way. The curved orbicularis oris does not work alone. Attached to it is a complex system of muscles that anatomists call the direct labial tractors, or DLT system. In humans, this system has evolved a unique configuration. The DLT muscles insert directly into the dermis, the deep layer of skin, and they are anatomically segregated from the orbicularis oris muscle itself. This separation is crucial. It means that the DLT muscles can pull and position the lips independently of the orbicularis oris, which provides opening, closing, and shaping functions. Think of it as having two separate control systems for the same structure, allowing for coordinated but independent movements. In other primates, the DLT architecture overlaps with the orbicularis oris. The muscles are not cleanly separated, but instead interdigitate and share attachment points. This overlapping architecture limits independent control. When a gorilla moves certain facial muscles, it affects multiple aspects of lip position simultaneously. The animal cannot as easily isolate and fine-tune individual components of lip movement. The human system, with its segregated DLT muscles, permits what engineers would call modular control. Each muscle can be activated in specific combinations, producing the refined motor control necessary for complex facial expressions and speech. The orbicularis oris muscle itself has a unique structural arrangement that sets it apart from most muscles in the body. Unlike typical skeletal muscles, which attach the bones at both ends, the orbicularis oris has no direct bony attachments whatsoever. Instead, it forms a floating muscular ring, suspended in place entirely by the other facial muscles that attach to it. This creates a remarkably flexible structure, free to move in ways that would be impossible if it were anchored to the skull. At the corners of the mouth, at points called the commissures, multiple facial muscles converge at a structure called the modiolus. This is a small knot of fibrous tissue where muscles from the cheeks, chin, and other parts of the face interdigitate with the orbicularis oris. The modiolus acts as an anchor point, allowing the various facial muscles to work in coordination. When you smile, muscles like the zygomaticus major pull the modiolus laterally and upward, which in turn stretches the orbicularis oris into that characteristic smile shape. When you frown, the depressor anguli oris pulls the modiolus downward, creating the opposite effect. The floating nature of the orbicularis oris, suspended by these converging muscles, is what gives the human mouth its extraordinary mobility and expressiveness. The fibers of the orbicularis oris do something unusual at the center of the upper lip. At the philtrum, that vertical groove running from the nose to the upper lip, the muscle fibers decussate, meaning they cross over to the opposite side. Fibers from the right side of the muscle cross over to insert into the left philtral ridge, and vice versa. This crossing pattern contributes to the complex shaping capabilities of the upper lip and helps create the distinctive Cupid's bow shape that characterizes the human upper lip margin. This decussation allows for even more refined control, as contractions on one side can influence the shape of the opposite side in coordinated ways. When the orbicularis oris contracts, it does not simply pull inward uniformly. The curved configuration means that different parts of the muscle can contract independently, creating localized shape changes. The upper lip can move separately from the lower lip. The corners of the mouth can be pulled in different directions than the center. This modularity of control is what allows for the incredible variety of human facial expressions. A smile involves pulling the corners of the mouth laterally and upward, while keeping the center relatively stable. A pout protrudes the lips forward while slightly everting them. A grimace pulls the lips taut and stretches them laterally. Each of these expressions requires precise, coordinated control of different segments of the orbicularis oris and the associated DLT muscles. The tissue composition of human lips reveals another layer of uniqueness. When researchers analyze the actual substance of lip tissue under microscopes, they find that human lips contain an unusually high proportion of adipose tissue and a remarkably low amount of connective tissue. Adipose tissue is fat, a soft and pliable substance. Connective tissue, by contrast, is tough and fibrous, providing structural support but limiting flexibility. The human lip is essentially built to be soft and malleable. Other primates show the opposite pattern. Chimpanzee lips, bonobo lips, and gorilla lips all contain much higher proportions of connective tissue and lower amounts of adipose tissue. The result is a firmer, less pliable structure. This might seem like a minor biochemical detail, but it has profound implications for function. The soft, adipose-rich human lip can be shaped and molded with minimal force. The muscles do not have to work as hard to reconfigure the lip into different positions. This reduces fatigue during extended use and permits rapid, repeated movements without strain. Consider what happens when you speak. Your lips move constantly, forming different shapes dozens of times per second. The word probability requires your lips to form a P sound at the beginning, round for the O sound, then quickly transition to the B sound, and finally shape themselves for the ending. This happens in less than a second. The soft, adipose-rich tissue allows these rapid transitions without requiring enormous muscular effort. A lip built primarily from tough connective tissue would resist these shape changes, making speech more effortful and less precise. The visible portion of the human lip, the reddish area that most people think of when they picture lips, is called the vermilion. This is the zone where the outer skin transitions to the mucous membrane of the mouth interior. The vermilion has its own unique properties. The skin here is extraordinarily thin, consisting of only 3 to 5 cellular layers, compared to the typical facial skin, which contains up to 16 layers. This thinness makes the vermilion almost translucent. Blood vessels beneath the surface show through, giving lips their characteristic reddish color in light-skinned individuals. In people with darker complexions, increased melanin in the lip skin makes this effect less pronounced, but the underlying vascular network remains the same. The thinness and high vascularization of the vermilion serve multiple functions. The rich blood supply keeps the tissue warm and metabolically active. It also makes the lips highly sensitive to temperature, pressure, and chemical stimuli. However, there is a trade-off. The thin vermilion lacks the protective features of normal skin. It has no hair follicles, no sweat glands, and no sebaceous glands that produce protective oils. This is why lips dry out so easily and why they are vulnerable to chapping in cold or dry weather. The border where the vermilion meets the normal facial skin is called the vermilion border, and it is outlined by a structure known as the white roll. This is a pale, slightly raised ridge formed by the bulging of the orbicularis oris muscle lying just beneath the skin. The white roll serves as a clear visual demarcation of the lip boundary. It is one of the features that makes human lips so visually prominent. In other primates, the transition from facial skin to the oral mucosa is far less distinct. There is no prominent vermilion, no clear white roll, and the lips blend more continuously into the surrounding face. The nerve supply to human lips is extraordinarily dense. The upper lip receives sensory innervation from the infraorbital nerve, a branch of the maxillary division of the trigeminal nerve. The lower lip is supplied by the mental nerve, which comes from the mandibular division of the trigeminal nerve. These nerves branch repeatedly within the lip tissue, creating a dense network of sensory receptors. The result is that lips are among the most sensitive parts of the human body. In the brain, the somatosensory cortex, the region responsible for processing touch sensations, devotes a disproportionately large area to the lips. If you were to draw a map of the body based on how much brain space is dedicated to each part, the lips would appear enormous, vastly out of proportion to their actual size. They would rival the hands in their neural representation, both structures dominating the somatosensory map far more than much larger body areas like the chest, back, or stomach. This reflects the functional importance of the lips. The brain allocates processing power where it is needed most, and the lips, like the hands, require enormous computational resources to support their precision and sensitivity. This extreme sensitivity serves multiple purposes. For infants, sensitive lips are crucial for feeding. Newborn humans are born with a sucking reflex, an automatic response where anything that touches the lips triggers sucking movements. This reflex is essential for survival, allowing the infant to breastfeed effectively. The lips must be sensitive enough to detect the breast and responsive enough to form a tight seal around it. Throughout life, the sensitivity of the lips allows us to explore textures and temperatures of food, detect harmful substances, and navigate our physical environment. We use our lips as tactile sensors, testing objects and surfaces in ways similar to how we use our fingertips. The implications of this anatomy extend to speech and facial expression. The curved orbicularis oris, the segregated DLT system, the adipose-rich tissue composition, and the floating muscular structure all work together to enable the rapid, precise movements required for human communication. No other primate possesses this combination of anatomical features, and no other primate can produce the range of sounds and expressions that humans routinely generate. This unique anatomy does come with biological costs. The prominent lips with their exposed vermilion are vulnerable to environmental damage and injury. The vertical orientation of the human face increases the risk of dental trauma. The anatomical reorganization that gives us these distinctive lips also affects other structures in the head and neck, creating trade-offs in terms of feeding efficiency and airway protection. Yet, despite these costs, the human lip anatomy has persisted and become universal across our species, suggesting that the benefits, whether in communication, social bonding, or other functions, have been substantial enough to outweigh the disadvantages. The human lip represents one of nature's most intricate mechanical systems, a structure where form and function converge to create capabilities found nowhere else in the animal kingdom. From the curved geometry of the orbicularis oris to the segregated control systems of the DLT muscles, from the soft adipose-rich tissue to the extraordinarily sensitive vermilion, every aspect of lip anatomy reflects a unique evolutionary path. This floating muscular ring, suspended without bony attachments and controlled by an elaborate network of muscles and nerves, stands as a testament to the remarkable complexity that can arise when evolutionary pressures favor precise motor control and sensitive perception. The human lip represents one of nature's most intricate mechanical systems, a structure where form and function converge to create capabilities found nowhere else in the animal kingdom. Chapter 2. Controversial aquatic ape hypothesis, suggesting waterside adaptation and slippery seafood consumption drove lip evolution. Among the many theories attempting to explain the peculiar anatomy of human lips, one stands out for both its explanatory power and its controversial status within the scientific community. This is the aquatic ape hypothesis, sometimes called the waterside hypothesis, which proposes that key human anatomical features evolved not on the African savanna, as traditional theories suggest, but along coastlines, riverbanks, and other aquatic environments, where our ancestors spent considerable time wading, swimming, and foraging for food in the water. The hypothesis is not new. It was first proposed in the 1960s and has been championed by writers like Elaine Morgan, who popularized the idea in several books. The central claim is straightforward. During a critical period of human evolution, likely in the Pleistocene era when archaic humans were dispersing along coasts and rivers, our ancestors adapted to a semi-aquatic lifestyle. This adaptation, proponents argue, explains a suite of unusual human features that are difficult to account for under conventional savanna-based evolutionary scenarios. Among these features are our hairless bodies, our subcutaneous fat layer, our ability to hold our breath, our bipedal posture, and, most relevant to this discussion, the unique structure of our mouths and lips. When you examine the human face in profile, one feature immediately distinguishes it from the faces of other primates. We have flat faces. Our mouths do not project forward in a snout or muzzle. Instead, they are positioned almost vertically beneath our eyes and nose. This retraction of the facial skeleton is called the absence of prognathicism. Chimpanzees, gorillas, and orangutans all have prognathic faces. Their jaws jut forward, creating a distinct snout. Human infants, interestingly, also have relatively flat faces. In fact, the human adult face retains many juvenile characteristics, a phenomenon called neoteny, where adult organisms retain features typical of their species' youth. Proponents of the aquatic ape hypothesis argue that this flat face may represent an adaptation for consuming slippery seafood. Think about the physical challenge of eating something like an oyster, a mussel, or a small fish. These foods are wet, slippery, and difficult to grasp. They do not require heavy chewing in the way that tough plant materials or raw meat might. Instead, they can be sucked into the mouth, manipulated briefly with the tongue, and swallowed. A flat face with lips positioned vertically beneath the nose creates an ideal configuration for this type of feeding. The lips can purse tightly to create suction, drawing slippery items into the oral cavity without the need for protruding jaws to bite and tear. Consider the alternative. A chimpanzee trying to eat a raw oyster would face considerable difficulty. The projecting jaws and relatively immobile lips would make it hard to create the tight seal necessary for effective suction. The oyster would slip and slide, difficult to control in position. For a human, the task is simple. Tilt your head back slightly, place the oyster shell against your lips, create a gentle suction, and the oyster slides smoothly into your mouth. No chewing is required. You can swallow it whole. This is not merely speculation. Humans have been harvesting and consuming shellfish for hundreds of thousands of years, and archaeological sites around the world contain massive shell middens, accumulations of discarded shells that testify to the importance of these foods in ancient diets. The structure of the human mouth reveals other features that seem suited for aquatic foraging. Inside your mouth, if you run your tongue along the roof, you will notice that the hard palate is smooth and arched. This is quite different from the palate structure in most other primates. Chimpanzees and other apes have relatively flat horizontal palates, marked by prominent transverse ridges called rugae palatini. These ridges serve a useful purpose for animals that eat foods requiring considerable chewing. The ridges help fix food in place, preventing it from slipping around during mastication. If you are eating tough plant material or fibrous meat, these ridges are helpful. They provide traction. But what if your diet consists largely of slippery, soft items that do not require chewing? In that case, the ridges become less useful, and a smooth palate offers different advantages. The human tongue is round, thick, and globular, quite unlike the long, flat tongue of a chimpanzee. This round tongue fits perfectly against the smooth, arched human palate. Together, they can seal off the oral cavity at virtually any point. The tongue can press against the palate at the front of the mouth, in the middle, or toward the back. This capability is essential for creating the kind of controlled suction necessary to draw small food items into the mouth, to manipulate them once inside, and to prevent water from entering unwanted spaces. This brings us to one of the most intriguing aspects of the aquatic ape hypothesis as it relates to lips. Humans possess an unusual ability among terrestrial mammals. We can feed underwater. Not many people realize this because it is not something we do in daily life, but the anatomy is there. You can submerge your face in water, open your mouth, and swallow food without inhaling water or allowing it to flood your throat and nasal passages. The coordination required is remarkable. Your tongue must position itself to close off the back of the oral cavity. Your soft palate must elevate to seal the nasal passage. Your lips must control water flow. And all of this must happen in precise sequence. This brings us to one of the most intriguing aspects of the aquatic ape hypothesis as it relates to lips. Humans possess an unusual ability among terrestrial mammals. We can feed underwater. Not many people realize this because it is not something we do in daily life, but the anatomy is there. You can submerge your face in water, open your mouth, and swallow food without inhaling water or allowing it to flood your throat and nasal passages. The coordination required is remarkable. Your tongue must position itself to close off the back of the oral cavity. Your soft palate must elevate to seal the nasal passage. Your lips must control water flow. And all of this must happen in precise sequence. This capability is rare in land animals but common in aquatic ones. Seals, sea lions, walruses, dolphins, and whales all feed underwater as a matter of routine. These animals have evolved specialized anatomy to manage the challenges of underwater feeding. Interestingly, many of them also have relatively small, rounded mouths and smooth palates. Walruses, which are specialist suction feeders, have blunt heads without protruding snouts. Their mouths are small and round. Globocephaline dolphins, another group of suction feeders, lack the elongated beaks characteristic of other dolphin species. Instead, they have rounded heads and compact oral openings. The parallels to human anatomy are striking. The human philtrum, that vertical groove running from the base of the nose to the center of the upper lip, also figures into the aquatic ape hypothesis. This feature is unique to humans among primates. Chimpanzees, gorillas, and orangutans do not have a distinct philtrum. In humans, the philtrum forms during embryonic development when three separate sections of tissue grow together to form the face. The central section descends from the area around the nose. The two lateral sections come from the sides. When they meet and fuse, they create the upper lip, and the philtrum marks the junction point where the central tissue merged with the lateral tissues. Advocates of the aquatic ape hypothesis suggest that the philtrum may relate to the overall reconfiguration of the human face for aquatic foraging. The precise mechanics are debated, but the structure itself is undeniable and requires explanation. One possibility is that the philtrum, along with the cupid's bow shape of the upper lip that it helps define, creates a more effective seal when the lips are pressed together. This seal would be important for preventing water from entering the mouth during underwater activity, or for creating the pressure differentials necessary for suction feeding. The small size of the human mouth opening is another feature that fits the aquatic scenario. When measured relative to body size, humans have much smaller mouth openings than other great apes. This is true even when controlling for our relatively large brains and heads. A smaller mouth opening is advantageous for suction feeding because it allows for the creation of greater negative pressure with less muscular effort. Think of it in mechanical terms. If you want to suck liquid through a straw, using a narrow straw allows you to create stronger suction than using a wide tube. The same principle applies to mouths. A smaller opening permits more effective suction with the same amount of lip and tongue movement. Some terrestrial animals do engage in suction feeding, but it is relatively rare and usually specialized. Certain primates, including some species of monkeys, can protrude their lips to suck juice from fruits or to extract grubs from tight spaces. Sloth bears use suction to feed on termites and ants. Some species of bats suck nectar from flowers. However, these animals still retain the basic primate facial structure of a relatively long, flat tongue and a ridged palate. Orangutans and chimpanzees can protrude their lips to create small mouth openings for sucking, but their overall oral anatomy differs fundamentally from that of humans. The human system appears optimized for suction in ways that go beyond what would be needed for occasional use on fruits or insects. We do not have to chew raw oysters. We can swallow small fish whole. We can open our mouths while swimming underwater without reflexively inhaling water. These capabilities suggest adaptation for regular, sustained interaction with aquatic foods, not occasional opportunistic consumption. During the Pleistocene, the epoch spanning from about 2.6 million years ago to roughly 12,000 years ago, archaic humans were indeed dispersing along coastlines and major river systems. Archaeological evidence shows human presence in coastal areas, and isotopic analysis of ancient human bones reveals signatures consistent with the consumption of seafood. Coastal and riverine environments would have offered rich and reliable food sources. Shellfish are abundant, easy to collect, and require no sophisticated hunting techniques. Fish can be caught in shallow water or trapped in tidal pools. These foods are also highly nutritious, rich in proteins and omega-3 fatty acids that are beneficial for brain development. If early humans were spending significant time foraging in shallow water, wading along shorelines, and consuming aquatic foods, the selective pressure for anatomical features that facilitated this lifestyle would have been considerable. A flatter face, smoother palate, round tongue, and mobile everted lips would all provide advantages. Over many generations, individuals with these features would have been more successful at exploiting aquatic resources, and the traits would have spread through the population. The ability to create controlled suction is not merely about getting food into the mouth. It also involves preventing water from going where it should not. When you are foraging in water, your face will inevitably get wet. Water will approach your nose and mouth. The ability to keep water out of your airways while still allowing you to manipulate food and swallow is critical. The human lip and tongue system, with its fine motor control and ability to seal off various passages, provides exactly this capability. The extremely flexible globular tongue works in concert with the closed parabolic tooth row and smooth arched palate to create a remarkably versatile system. The tongue can close the oral cavity at the dental arch, preventing material from passing the teeth. It can seal against the palate further back, closing off the throat. It can move laterally to direct food and liquid to specific areas. This modularity of control is precisely what would be needed for underwater feeding, where the consequences of aspiration could be immediately fatal. Critics of the aquatic ape hypothesis raise several objections. The most common is that the hypothesis is not parsimonious. In other words, it requires proposing a specific environmental scenario for which the fossil evidence is incomplete. Traditional savanna-based theories of human evolution are supported by extensive paleontological and archaeological data showing early humans in grassland and mixed woodland environments. While there is evidence of coastal and riverine occupation, critics argue that this does not necessarily indicate a semi-aquatic lifestyle significant enough to drive major anatomical changes. Another objection concerns the fact that other features predicted by the aquatic ape hypothesis are less well supported. For example, the hypothesis suggests that human hairlessness and subcutaneous fat are aquatic adaptations, similar to those seen in seals and whales. However, other explanations for these features exist, including thermoregulatory advantages in hot climates and energy storage for brain development. The hypothesis has been accused of cherry-picking features that fit the aquatic scenario while ignoring other aspects of human anatomy that do not. Despite these criticisms, the aquatic ape hypothesis has proven remarkably persistent. Part of its appeal lies in its ability to account for features that are otherwise puzzling. Why do humans have such unusual mouths compared to other apes? Why can we perform suction feeding so effectively? Why do we have smooth palates and round tongues? Traditional theories often struggle to provide satisfying answers to these questions. The aquatic hypothesis, whether fully correct or not, offers a coherent explanatory framework. It is worth noting that the hypothesis need not be an all-or-nothing proposition. It is entirely possible that some human anatomical features evolved in response to aquatic or semi-aquatic behaviors without humans having been fully aquatic creatures. Our ancestors may have exploited coastal and riverine resources extensively during certain periods or in certain populations, and this could have been sufficient to drive the evolution of specific adaptations, like lip structure, even if they spent most of their time on land. The connection between lip anatomy and speech, which will be explored in greater depth in the next chapter, also figures into this discussion. If lips first evolved enhanced motor control for the purpose of suction feeding on slippery seafood, that same motor control could later have been co-opted for speech production. Evolution frequently repurposes existing structures for new functions. The wings of birds evolved from the forelimbs of dinosaurs, which originally served entirely different purposes. Similarly, the lips that once helped our ancestors extract oysters from shells may have later become the articulators that allowed us to produce the complex sounds of human language. The question of why the aquatic ape hypothesis remains controversial despite its explanatory power is itself interesting. Part of the answer lies in the sociology of science. The hypothesis was popularized by non-specialists and has often been presented in popular rather than academic contexts. This has led some researchers to dismiss it without thorough evaluation. Additionally, the hypothesis challenges long-standing narratives about human evolution, narratives that have been taught in textbooks and reinforced through decades of research. Paradigm shifts in science are always difficult, even when new evidence emerges. What is clear is that the human lip anatomy requires explanation. Whether that explanation ultimately involves waterside adaptation and seafood consumption, or whether other factors prove more important, remains an open question. The anatomy itself, however, is not in dispute. We have small mouths, smooth palates, round tongues, flat faces, and uniquely mobile lips. These features distinguish us from all other primates, and indeed from most terrestrial mammals. The aquatic ape hypothesis, controversial though it may be, provides one possible answer to the question of how and why we came to possess these distinctive traits. Chapter 3. Evolution of Speech from Primate Lip Smacking to Complex Human Articulation and Language. If you have ever watched a mother monkey with her infant, you may have noticed something curious. The mother will often make rapid smacking movements with her lips while looking directly at her baby's face. The movement is quick, rhythmic, and repetitive. It produces a soft popping sound, something like pages. This behavior, called lip smacking, appears in many primate species, and for a long time it was understood simply as a friendly social gesture, a way of communicating affection or establishing calm in close interactions. What no one suspected until quite recently is that this simple behavior might hold the key to understanding how human speech evolved. The breakthrough came when researchers at Princeton University and the University of Vienna used high-speed X-ray cinematography to analyze exactly what happens inside a monkey's mouth during lip smacking. What they discovered was astonishing. Lip smacking is not just a simple opening and closing of the lips. It is an extraordinarily complex behavior requiring the rapid, coordinated movement of the lips, jaw, tongue, and even the hyoid bone, the small horseshoe-shaped bone that supports the larynx and tongue. All of these structures must move in precise synchronization, and they do so at a very specific rate. approximately 5 cycles per second. This number is significant. 5 cycles per second is exactly the rate at which humans produce speech syllables. When you speak a sentence, your articulators, the parts of your mouth and throat that shape sound, move at this same fundamental rhythm. It is much faster than the movements involved in chewing, which occur at only about 2.5 cycles per second. The match between lip-smacking speed and speech speed is too precise to be coincidental. It suggests that the neural and muscular systems controlling lip-smacking in monkeys may be evolutionarily related to the systems that control speech in humans. This discovery lends powerful support to a hypothesis first proposed decades ago by researcher Peter McNelage. He suggested that the origins of human speech might not lie primarily in primate vocalizations, the hoots and caws that monkeys and apes use to communicate, but rather in their facial gestures. The idea was initially met with skepticism. After all, speech is fundamentally about sound. How could silent or nearly silent facial movements be the foundation for spoken language? The answer lies in understanding what speech actually requires. Yes, speech involves sound production through phonation, the vibration of the vocal cords in the larynx. But phonation alone does not create intelligible speech. If you were to simply vibrate your vocal cords without moving your tongue, lips, or jaw, you would produce only a monotonous hum. What makes speech comprehensible is the rapid, precise modulation of that sound by the structures of the vocal tract. And the most critical of those structures are the lips. When you speak, you are constantly forming and releasing constrictions at various points in your mouth and throat. The alternation between vowels and consonants that creates the rhythm of speech syllables depends on these rapid movements. A consonant like p or b requires you to completely close your lips, build up air pressure behind them, and then release it explosively. A vowel like or e requires you to open the mouth and shape the vocal tract in a specific way without creating a complete closure anywhere. The cycle of closing and opening, consonant and vowel, closure and release is precisely what creates the syllable structure of human language. Now look again at monkey lip smacking. The movement pattern is strikingly similar. The lips come together, creating a closure. Then they separate, creating an opening. This happens rapidly and rhythmically. Although the behavior does not involve phonation in monkeys, it would take only a relatively small evolutionary step to add voicing to these movements. If the larynx were activated during lip smacking, producing sound while the lips opened and closed, the result would be something very much like human speech syllables. The evidence goes beyond macaques. Chimpanzees, our closest living relatives, also make communicative sounds with their lips. They produce loud lip smacks similar to those of monkeys, but they also create what researchers call lip buzzes, or what most people would recognize as raspberries. These are sounds produced by forcing air through loosely closed lips, causing them to vibrate. Unlike the innate vocalizations that chimpanzees produce from the larynx, these lip-based sounds appear to be under voluntary control. Chimpanzees can learn to make them, and they use them deliberately in social contexts. This distinction between voluntary and involuntary vocalizations is crucial. Most primate calls are innate. A chimpanzee does not learn to make a food call or an alarm call. These vocalizations are hardwired into the nervous system and are produced automatically in response to specific stimuli. They are similar to human laughter or crying, which are also largely involuntary. But speech is different. Speech is learned. Every human child must learn the specific sounds and words of their particular language through years of practice and social interaction. The fact that chimpanzee lip sounds are voluntary and learned suggests that the neural systems controlling them are different from those controlling innate vocalizations. They may represent an evolutionary intermediate between fixed, instinctive calls and fully flexible, learned speech. The same pattern appears in orangutans, which can learn to whistle, producing sound by shaping their lips and controlling airflow without using the larynx. Again, this is a learned, voluntary behavior, not an instinctive one. If the motor control systems for lip movements in primates provided the foundation for speech, then the evolution of human language required two major changes. First, the voluntary control over lip and facial movements had to become linked to vocalization, so that sound was produced simultaneously with the lip movements rather than separately. Second, the anatomy of the human vocal tract had to change in ways that permitted a much wider range of sounds to be produced. The same pattern appears in orangutans, which can learn to whistle, producing sound by shaping their lips and controlling airflow without using the larynx. Again, this is a learned, voluntary behavior, not an instinctive one. If the motor control systems for lip movements in primates provided the foundation for speech, then the evolution of human language required two major changes. First, the voluntary control over lip and facial movements had to become linked to vocalization, so that sound was produced simultaneously with the lip movements rather than separately. Second, the anatomy of the human vocal tract had to change in ways that permitted a much wider range of sounds to be produced. The anatomical changes were substantial. As you have already learned, human lips have a unique, curved muscular structure that permits extraordinarily precise movements. But lips are only part of the system. The entire vocal tract had to be reconfigured. In adult humans, the vocal tract is divided into two sections of approximately equal length. The horizontal section runs through the mouth, from the lips to the back of the oral cavity. The vertical section runs down through the pharynx, the space behind the tongue and above the larynx. These two sections meet at roughly a right angle. This configuration is unique to adult humans. All other primates and even human infants have vocal tracts that are proportionally much longer in the horizontal dimension and much shorter in the vertical one. The equal length configuration of the adult human vocal tract has a specific acoustic consequence. It allows the tongue, positioned at the junction of the two tubes, to create dramatic changes in the shape of the resonating space. By moving the tongue body forward or backward, or by raising or lowering it, a speaker can produce tenfold changes in the cross-sectional area of the vocal tract. These abrupt changes in shape are what create the distinctive acoustic signatures of certain vowels. Specifically, the human vocal tract can produce what phoneticians call quantovowels. These are vowel sounds whose acoustic properties remain stable even if the articulation is not perfectly precise. The three most important quantovowels are the I sound in the word see, the U sound in the word do, and the A sound in the word ma. These three vowels appear in virtually every human language because they are acoustically distinct and easy for listeners to tell apart. They occupy widely separated regions in the acoustic space of possible vowel sounds. If you make a small error in tongue position while producing an I sound, it still sounds like I and not like some other vowel. The quantovowels are robust and reliable. No other primate can produce these sounds. The disproportion between the horizontal and vertical dimensions of their vocal tracts makes it physically impossible for them to achieve the necessary tongue positions. This is not a matter of practice or learning. A chimpanzee could not learn to say the word see no matter how much training it received, because its vocal tract simply cannot create the required sound. The limitation is anatomical, not cognitive. The human vocal tract configuration comes at a steep cost. The descent of the larynx into the throat, which creates the elongated vertical portion of the vocal tract, dramatically increases the risk of choking. In most mammals, the larynx is positioned high in the neck, and it can lock into the back of the nasal cavity during swallowing. This creates two separate tubes, one for breathing and one for swallowing, allowing the animal to breathe and swallow at the same time. Human infants have this arrangement for the first months of life, which is why they can nurse and breathe simultaneously. But as the larynx descends during childhood, this safety mechanism is lost. In adult humans, food and liquid must pass over the opening to the windpipe on their way to the esophagus. If the timing is wrong, if the epiglottis fails to seal the airway at precisely the right moment, material can enter the lungs. Choking is a major cause of accidental death in humans, killing hundreds of thousands of people worldwide every year. For evolution to have favored an anatomical change that carries such a serious survival cost, the benefit must have been enormous. The most plausible benefit is enhanced communication through speech. If the ability to produce clear, rapid, acoustically distinct sounds provided a significant advantage in social coordination, in teaching, in planning, and in transmitting knowledge, then the risk of choking might be outweighed by the advantage of effective vocal communication. But anatomy alone does not explain speech. the brain had to change as well. Research in neuroscience over the past several decades has revealed that the neural systems responsible for speech are not localized in a single region of the brain, as was once thought. Instead, speech depends on circuits, loops of neural connections that link different brain regions together. Interestingly, many of these same circuits are involved in motor control in other animals. In humans, however, these motor control circuits have been recruited for additional functions, including the regulation of speech, the processing of complex syntax, and even certain aspects of abstract thought. This has a profound implication. It suggests that the evolution of the brain systems necessary for language may have been driven, at least in part, by the increasing demands of speech motor control. As our ancestors developed more sophisticated vocal communication, they needed to exert finer and finer control over the muscles of the lips, tongue, jaw, and larynx. This created selection pressure for enhanced motor control circuits in the brain. But once these enhanced circuits existed, they could be co-opted for other purposes. A brain that can coordinate the rapid, precise movements necessary to produce the word probability in less than 1 second is a brain with significant computational power. That same power can be applied to sequencing thoughts, manipulating abstract concepts, and planning complex actions. In other words, we may have talked ourselves into being smarter. The evolutionary feedback loop between speech and cognition could have worked as follows. Initially, enhanced motor control of the lips and vocal tract allowed for clearer, faster communication. This was advantageous, so individuals with better motor control had more offspring. But the neural circuits that provided better motor control also had the side effect of enhancing cognitive flexibility. Individuals who could not only speak clearly but also think more flexibly had an even greater advantage. Over many generations, this process would drive the expansion and reorganization of the brain, producing the distinctive human pattern of large brains with highly developed frontal and temporal regions. The timeline of this evolutionary process is still debated, but evidence from multiple sources points to a relatively recent emergence of fully modern speech capabilities. Genetic, anatomical, and archaeological data suggest that the specialized anatomy and neural mechanisms underlying fully human speech, language, and advanced cognitive abilities reached their present state sometime between 100,000 and 50,000 years ago. This is remarkably recent. Our species, Homo sapiens, originated roughly 200,000 years ago. For the first half of our species' existence, we may have had modern-looking bodies, but not yet fully modern speech and cognition. This has important implications for how we understand human evolution. It suggests that there was a period during which anatomically modern humans existed, but had not yet developed the full suite of behavioral capabilities that we associate with modern humanity. They may have had speech, but perhaps not the full range of sounds and the full speed and precision of articulation that we possess today. They may have had language, but perhaps with simpler syntax and smaller vocabularies. They may have had culture and symbolic thought, but perhaps less elaborate and sophisticated than what emerged later. What changed between 100,000 years ago and 50,000 years ago? We cannot be certain, but the evidence points to incremental improvements in speech motor control and the cascading effects those improvements had on language and cognition. Small changes in vocal tract anatomy, perhaps driven by genetic mutations affecting facial development, would have allowed for the production of clearer vowels. This would have made communication more efficient, creating selection pressure for individuals who could take advantage of these new sounds. Simultaneously, changes in brain circuitry that enhanced motor control would have improved the speed and precision with which speech sounds could be produced. This would have allowed for faster speech, which in turn would have allowed for the transmission of more information in less time. As speech became faster and more precise, the demands on listeners would have increased as well. To understand rapid speech, you must be able to segment the continuous acoustic stream into discrete phonemes, syllables, and words. You must hold the beginning of a sentence in working memory while processing the end. You must extract syntactic structure from word order and grammatical markers. All of these tasks require sophisticated neural processing. The evolution of better speech would have created selection pressure for better listening and comprehension abilities, which in turn would have driven further brain evolution. The result of this evolutionary arms race between speaking and listening is the modern human communication system, a system of staggering complexity and efficiency. When you speak, you produce somewhere between 10 and 15 speech sounds per second. Each sound is created by a specific configuration of your lips, tongue, jaw, and larynx, and each configuration must be achieved with precision and held for only a fraction of a second before transitioning to the next. The acoustic signal produced is enormously complex, with multiple overlapping acoustic cues that listeners use to identify phonemes. Yet this process, which would seem impossibly difficult if you had to think about it consciously, happens automatically and effortlessly. This automaticity is the signature of a deeply evolved system. The neural circuits controlling speech operate largely below the level of conscious awareness. You do not think about where to place your tongue to make a t sound or how much to round your lips for an o sound. You simply think the thought you want to express, and the words emerge fully formed from your mouth. The fact that this can happen at all is a testament to millions of years of evolutionary refinement, beginning with the simple lip smacking of a monkey and culminating in the uniquely human capacity for articulate speech. The story of speech evolution is also a story about the relationship between form and function. The unique anatomy of human lips did not evolve in a vacuum. It evolved in concert with changes in the tongue, the vocal tract, the larynx, and the brain. Each change opened up new functional possibilities, and each new function created selection pressure for further anatomical refinement. The curved orbicularis oris muscle that gives human lips their distinctive shape is not just a curious anatomical detail. It is an essential component of a larger system that allows humans to communicate in ways that no other species can match. And it all may have started with a monkey making soft popping sounds to comfort her infant, a simple gesture of affection that contained, hidden within it, the seeds of human language. Chapter 4. The 21 Million Year Kiss. When you kiss someone, you are engaging in a behavior whose origins stretch back through time to an era long before humans existed. The evolutionary history of kissing extends across millions of years, connecting us to ancestors we share with chimpanzees, bonobos, orangutans, and gorillas. A 2024 study led by evolutionary biologist Matilda Brindle at Oxford's Department of Biology, published in the journal Evolution and Human Behavior, traced this intimate behavior back approximately 21 million years, revealing that kissing is not a modern human invention, but an ancient trait inherited from the common ancestor of all great apes. This discovery emerged from a detective story of sorts, one that required researchers to work backward through evolutionary time using clues from living animals. The challenge was formidable. Unlike bones, teeth, or other physical structures that can fossilize and be excavated from ancient sediments, behavior leaves no direct trace in the geological record. You cannot dig up a fossilized kiss. Instead, scientists had to employ a different approach, one that relies on reconstructing the evolutionary relationships among living species and then using statistical models to infer what behaviors their extinct ancestors likely possessed. The research team collected observations from existing scientific literature documenting kissing behavior across the primate world. They defined kissing specifically as non-aggressive, mouth-to-mouth contact that does not involve the transfer of food. Using this definition, they found that several living primate species engage in kissing. Chimpanzees kiss, bonobos kiss, sometimes with considerable tongue involvement. Orangutans have been observed kissing. Even one species of gorilla engages in the behavior. These observations provided the raw data for the analysis. The next step involved constructing what is called a phylogenetic tree, a branching diagram that represents the evolutionary relationships among species based on their genetic similarities and differences. Think of it as a family tree, but one that extends back millions of years and includes not just individuals but entire species. The tree shows how different primate lineages diverge from common ancestors over time. Chimpanzees and bonobos are closely related to each other and share a relatively recent common ancestor. Humans are closely related to chimpanzees and bonobos, with all three species sharing a common ancestor that lived about 6 to 7 million years ago. Orangutans diverged from the lineage leading to African apes and humans much earlier, perhaps 14 to 16 million years ago. Gorillas occupy an intermediate position in the tree. With this phylogenetic framework in place, researchers could apply statistical modeling techniques. The logic is straightforward. If multiple species descended from a common ancestor all share a particular trait, it is likely that the common ancestor possessed that trait as well. For example, if chimpanzees, bonobos, and humans all kiss, and if these three species share a common ancestor, then that common ancestor probably kissed too. The trait was inherited, passed down through the generations as each lineage evolved separately. The modeling process is sophisticated. It does not simply assume that shared traits must have been inherited. Instead, it calculates probabilities. It considers multiple scenarios, including the possibility that kissing evolved independently in different lineages rather than being inherited from a common ancestor. It weighs the evidence, comparing different evolutionary pathways to determine which is most likely given the observed data. To ensure robust results, the researchers ran their statistical models 10 million times, each time slightly varying the parameters to test the stability of their conclusions. The results pointed consistently to a single conclusion. Kissing is an evolutionarily ancient behavior in great apes. It did not evolve separately in different lineages. Instead, it appeared once in a common ancestor and has been retained, to varying degrees, in the descendant species. The timing of this ancestral origin can be estimated based on when the different ape lineages diverge from one another. Because orangutans kiss, and because orangutans separated from the African ape and human lineage somewhere between 14 and 16 million years ago, the behavior must have already existed in the common ancestor of all these groups. The statistical models place the origin of kissing between 21.5 million and 16.9 million years ago. This is a staggering length of time. 21 million years ago, the world was a very different place. The continents occupied slightly different positions, the climate was warmer, the landscape of what would later become Africa was covered in tropical forests, and somewhere in those forests lived the ancestor of all modern great apes, a creature that looked nothing like a modern human, but that nevertheless engaged in the intimate behavior of pressing its lips against the lips of others. The implications extend to extinct human relatives. Neanderthals, who evolved separately from the lineage leading to modern humans starting about 500,000 years ago, were still members of the broader human family. If the common ancestor of all great apes kissed, and if that behavior was retained in the lineages leading to modern humans, then it was almost certainly retained in the Neanderthal lineage as well. Neanderthals, though stocky, cold-adapted humans who lived across Europe and Western Asia until roughly 40,000 years ago, likely kissed each other. They may have kissed their children, they may have kissed their mates. The behavior would have been as natural to them as it is to us. Even more intriguingly, there is genetic evidence that modern humans and Neanderthals interbred when their populations encountered each other. If Neanderthals and modern humans were mating, producing offspring together, it is entirely plausible that they were also kissing. The image is striking: a modern human and a Neanderthal, two separate branches of the human family tree that had evolved independently for hundreds of thousands of years, reunited and engaging in one of the most intimate human behaviors. The implications extend to extinct human relatives. Neanderthals, who evolved separately from the lineage leading to modern humans starting about 500,000 years ago, were still members of the broader human family. If the common ancestor of all great apes kissed, and if that behavior was retained in the lineages leading to modern humans, then it was almost certainly retained in the Neanderthal lineage as well. Neanderthals, though stocky, cold-adapted humans who lived across Europe and Western Asia until roughly 40,000 years ago, likely kissed each other. They may have kissed their children. They may have kissed their mates. The behavior would have been as natural to them as it is to us. Even more intriguingly, there is genetic evidence that modern humans and Neanderthals interbred when their populations encountered each other. If Neanderthals and modern humans were mating, producing offspring together, it is entirely plausible that they were also kissing. The image is striking: a modern human and a Neanderthal, Two separate branches of the human family tree that had evolved independently for hundreds of thousands of years, reunited and engaging in one of the most intimate human behaviors. The phylogenetic analysis reveals with considerable confidence when kissing evolved. What it does not reveal is why. The question of ultimate causation, the specific selective pressures that drove the initial evolution of this behavior more than 20 million years ago, remains elusive. Kissing presents what researchers have termed an evolutionary conundrum. The behavior carries obvious risks. When you press your mouth against another person's mouth, you are exchanging saliva, mucus, and the microorganisms that live in both. You are creating a direct pathway for the transmission of infectious diseases. Bacteria and viruses can pass easily from one individual to another during a kiss. Throughout human history, countless people have contracted illnesses from kissing. In an era before modern medicine, some of these illnesses would have been fatal. Against this cost, what is the benefit? Unlike behaviors that clearly enhance survival or reproduction, such as eating, mating, or caring for offspring, kissing does not have an obvious function that would outweigh its risks. You do not need to kiss in order to reproduce. You can raise children perfectly well without ever kissing them. You can form social bonds through other means. So why would evolution have favored a behavior that is dangerous and seemingly unnecessary? The answer may lie in the fact that kissing serves not one function, but many. Among primates, kissing has been observed in a variety of contexts, each suggesting a different purpose. Chimpanzees kiss after fights, pressing their lips together as a gesture of reconciliation. The behavior appears to reduce tension and restore calm relationships after conflict. This suggests that kissing may function as a signal of peaceful intent, a way of communicating that aggression has ended and cooperation can resume. Bonobos, known for their highly sexual and affiliative social system, use kissing in contexts that appear related to bonding and affiliation. They kiss frequently, and the behavior often involves considerable tongue contact. For bonobos, kissing seems to be part of a broader repertoire of behaviors that maintain social cohesion in their groups. In humans, kissing appears in an even wider range of contexts. Parents kiss children as an expression of care and affection. This behavior begins in infancy and continues throughout life, serving to strengthen the emotional bond between parent and child. Friends may kiss as a greeting or farewell, a ritualized expression of friendship. Romantic partners kiss as part of courtship and sexual activity, where it serves both to signal interest and to assess compatibility. The assessment function is particularly interesting. Gordon Gallup's research on American college students found that 59% of men and 66% of women reported having felt attracted to someone until they kissed them, at which point the attraction disappeared. The kiss revealed chemical information about immune system compatibility, hormonal profiles, or other biological factors that visual appearance alone had not conveyed. And that information proved disqualifying. The person was no longer desirable as a mate. This suggests that kissing functions as a filter, a way of screening potential partners for compatibility. The lips' extraordinary sensitivity, with their dense nerve endings and disproportionate brain representation detailed in Chapter 1, makes them ideal instruments for gathering both tactile and chemical information during a kiss. Human saliva contains a complex mixture of substances, including hormones, proteins, and other molecules that can provide information about a person's health, genetic makeup, and current physiological state. When you kiss someone, you are tasting their saliva. The taste receptors in your mouth and the olfactory receptors in your nose analyze the chemical composition, and this information is processed by brain regions involved in emotion and decision-making. The process is entirely unconscious, but it influences your feelings about the person. By rejecting partners whose chemical signals are unfavorable, individuals can avoid investing time and resources in relationships unlikely to produce healthy offspring. Interestingly, the importance of kissing in mate assessment appears to vary by gender. Research indicates that women place greater emphasis on kissing when evaluating potential partners than men do. Women report being less likely to have sex with someone they have not kissed, and they are more likely to end a potential relationship if the first kiss is unsatisfactory. This gender difference may relate to the greater biological investment that women make in reproduction. For a woman, a poor mate choice can result in 9 months of pregnancy, the risks of childbirth, and years of intensive parental care for a child who may have inherited unfavorable genetic traits. For a man, the minimum biological investment in reproduction is far smaller. Therefore, women may have evolved to be more selective, and kissing may be one of the tools that enables that selectivity. The exchange of chemical information during kissing goes beyond taste. Scent also plays a role. Although humans have a relatively poor sense of smell compared to many other mammals, we can still detect airborne molecules released from another person's skin and breath. During a kiss, the close proximity of two faces ensures that each person is inhaling the other's scent. Some of the molecules being detected may be pheromones, chemical signals that unconsciously influence behavior and physiology. The existence and importance of human pheromones remains debated, but regardless of whether specific pheromone molecules are involved, it is clear that scent contributes to the overall assessment of a partner's suitability. The reddish color of lips, resulting from blood vessels visible through thin skin as described in Chapter 1, also functions as a fertility signal. This visual cue, combined with the chemical and tactile information exchanged during kissing, provides a multi-sensory assessment system for evaluating potential mates. This chemical signaling function may help explain why kissing is not universal across human cultures, despite its deep evolutionary roots. While the majority of human societies practice some form of romantic or sexual kissing, a significant minority do not. Surveys have found that kissing is documented in only about 46% of cultures worldwide. Charles Darwin himself noted this variation in the 19th century, listing cultures where mouth-to-mouth kissing was absent or rare, including the Maori, Tahitians, Papuans, Australians, Somals of Africa, and Eskimo. In the cultures where mouth-to-mouth kissing is absent, people engage in alternative behaviors that may serve similar functions. Partners may blow in each other's faces, lick each other's skin, suck on each other's lips or tongues, or rub faces together. These behaviors also permit the exchange of chemical information and the close sensory evaluation of a partner. The practice, often described as an Eskimo kiss, where two people rub noses together, is actually not primarily about tactile contact between noses. Early Arctic explorers who first described the behavior misunderstood what they were observing. The behavior is more accurately described as sniffing, an exchange of scents in which each person inhales deeply while holding their face close to the others. This serves the same assessment function as kissing, but through a different sensory modality, emphasizing smell over taste and touch. The fact that kissing is not universal, despite being evolutionarily ancient, reveals something important about the relationship between biological inheritance and cultural practice. Just because a behavior has deep evolutionary roots does not mean it must be expressed in all populations. Primates, including humans, are extraordinarily flexible and intelligent. They can modify their behavior based on learning, cultural transmission, and local circumstances. A behavior that is useful in one context may be less useful or even harmful in another. Kissing carries genuine risks. The transmission of infectious disease is a serious concern, particularly in populations without modern medicine or in situations where epidemic diseases are present. In such contexts, cultural norms that discourage mouth-to-mouth contact might arise and persist because they reduce disease transmission. Conversely, in contexts where the benefits of kissing, such as enhanced social bonding and mate assessment, outweigh the risks, the behavior would be retained and perhaps even elaborated into complex cultural rituals. Another function of kissing that may contribute to its evolutionary persistence is anxiety reduction. This hypothesis, proposed by researchers including Chip Walter in Scientific American Mind and evolutionary biologist Desmond Morris, draws on observations of primate food-sharing behavior. As detailed in Chapter 3, chimpanzee mothers sometimes chew food and then transfer it directly from their mouths to the mouths of their infants by pressing their lips together. The pairing of lip contact with the receipt of food could create a conditioned association through basic classical conditioning. Over time, the lip contact alone, even without the transfer of food, could trigger feelings of comfort and satisfaction. This might explain why pressing lips together, even in contexts unrelated to feeding, feels pleasant and reduces anxiety. The behavior has been co-opted from its original nutritional function and now serves a purely social and emotional function.
When you kiss someone you care about, whether a child, a friend, or a romantic partner, you are engaging in a behavior with multiple layers of meaning and function. On the surface, it is a simple physical act, the pressing of lips against lips. But beneath that surface lie millions of years of evolutionary history, complex neurological responses, unconscious chemical assessments, and profound emotional significance. The behavior simultaneously serves to bond, to soothe, to arouse, and to evaluate. It is both deeply personal and fundamentally biological, both learned and innate. The phylogenetics detective work has established the timing. Kissing evolved more than 20 million years ago in the common ancestor of all great apes. It has traced the behavior's presence across the primate family tree and inferred its likely presence in extinct human relatives, including the Neanderthals. Researchers have identified multiple functions kissing serves in living species, from conflict resolution to mate assessment to anxiety reduction. But the fundamental question of why this particular behavior, with its peculiar combination of intimacy and risk, first evolved remains unanswered. The selective pressures that favored lip-to-lip contact in that ancient ancestor, the specific ecological or social circumstances that made kissing advantageous enough to outweigh its costs, are still matters of speculation rather than established fact. That deeper mystery awaits future investigation, future evidence, and future insights into the lives of our distant ancestors who first pressed their lips together in what would become one of humanity's most recognizable and cherished gestures.
Chapter 5. The Fertility Signal. When you look at another person's face, you are unconsciously processing an enormous amount of information. Within fractions of a second, your brain makes assessments about age, health, emotional state, and, whether you realize it or not, reproductive potential. Among the features your brain evaluates, the lips occupy a position of surprising importance. The size, shape, color, and fullness of lips function as biological signals, conveying information about hormone levels, fertility, and genetic quality. This is not a modern cultural invention. It is an ancient aspect of human mate selection, shaped by hundreds of thousands of years of sexual selection. Sexual selection is a specific form of natural selection that operates through competition for mates and mate choice. It is the force that gave peacocks their elaborate tail feathers, male lions their impressive manes, and deer their massive antlers. In humans, sexual selection has shaped many aspects of our bodies, from the distribution of body hair to the pitch of our voices. And it has certainly shaped our lips. The connection between lip fullness and fertility centers on a hormone called estrogen. Estrogen is one of the primary female sex hormones, responsible for the development and regulation of the female reproductive system and secondary sexual characteristics. During puberty, as estrogen levels rise in girls, the body undergoes a series of changes. The breasts develop, the hips widen, fat is redistributed to create a more feminine body shape, and the face changes in subtle but important ways. One of estrogen's effects on the face is to maintain what researchers describe as a relatively childlike and youthful structure. While testosterone in males causes the face to become more angular, with a heavier brow, a larger jaw, and more prominent bone structure overall, estrogen works in the opposite direction. It preserves softer contours, it keeps the eyes proportionally larger, and it maintains fuller lips. Research in evolutionary psychology has documented this relationship with considerable precision. Studies measuring the hormone levels of women during puberty and comparing those levels to facial measurements have found a clear correlation. The more estrogen a woman produces, the larger her eyes appear relative to her face, and the fuller her lips become. These features are not arbitrary. They are honest biological signals, difficult to fake without artificial intervention, that communicate real information about a woman's hormonal status. Why does this matter for reproduction? Estrogen levels are closely tied to fertility. Women with healthy estrogen production are more likely to ovulate regularly, to maintain pregnancy successfully and to have healthy offspring. In the ancestral environment, before modern medicine, these factors were critical determinants of reproductive success. A man choosing a mate was, whether he knew it or not, making a decision that would determine whether his genes were passed to the next generation. Features that reliably indicated high fertility would have been strongly preferred, and over many generations, this preference would have become hardwired into male psychology.
The result is that men across cultures show a consistent preference for fuller lips in women. This is not something that varies randomly from place to place or era to era. Survey research conducted by psychologists studying human attraction has found that the preference for full lips transcends cultural boundaries. Whether the research subjects are in Europe, Asia, Africa, or the Americas, whether they are from traditional societies or modern urban environments, men consistently rate women with fuller lips as more attractive than women with thinner lips. The preference is not absolute. Extreme lip fullness, whether natural or artificially enhanced, can cross a threshold and become unattractive. But within the normal range of human variation, fuller is generally perceived as more attractive. This universality is a strong indicator that the preference has biological rather than purely cultural origins. The attractiveness of full lips is not static across a woman's lifespan. As women age, their lips change in predictable ways. Collagen is a structural protein that provides firmness and volume to skin and other tissues. Young skin produces collagen abundantly, but collagen production declines with age, beginning as early as the mid-20s and accelerating after menopause, when estrogen levels drop sharply. As collagen decreases, the lips lose volume. They become thinner. The vermilion border, that crisp line where the reddish lip tissue meets the surrounding skin, becomes less defined. The Cupid's bow, the distinctive double curve at the center of the upper lip, flattens. These changes are visible and noticeable. Research tracking lip appearance across the lifespan has found that lips become visibly aged starting in the 40s, with the changes becoming increasingly pronounced in the 50s and beyond. The thinning is progressive. What was once a full, voluptuous lip in a 20-year-old woman becomes a thin, flat structure by age 70. The biological meaning of this change is clear. Thinning lips signal declining fertility. A woman in her 20s with full lips is at the peak of her reproductive years. A woman in her 60s with thin lips is postmenopausal, no longer capable of bearing children. From an evolutionary perspective, this creates a direct visual link between lip appearance and reproductive value. Men who were preferentially attracted to women with full lips would have been, on average, choosing women who were younger and more fertile. Those men would have had more offspring, and the genes underlying the preference would have spread through the population. Over thousands of generations, the preference would have become universal. But sexual selection does not operate in only one direction. Women also choose mates, and their choices exert selective pressure on male traits. Interestingly, research has found that lip size also plays a role in female attraction to men, though the pattern is somewhat different. Women do not prefer extremely full lips in men, nor do they prefer very thin lips. Instead, the preference is for what researchers describe as masculine lips that are middle-sized, rugged, and sensual. The lips should be well-defined with a clear shape, but not overly plump. This makes biological sense. In men, extremely full lips might signal high estrogen levels, which would not be advantageous for male reproductive success. Testosterone, not estrogen, is the key hormone for male fertility and sexual function. A man's face should signal high testosterone, which typically means a more angular jaw, a heavier brow, and moderately sized, well-defined lips rather than extremely full ones. The actor and musician known as LL Cool J provides a notable example. His lips are full by male standards, contributing to his distinctive appearance, but they are balanced with other masculine facial features. The result is perceived as attractive, a combination of softness and strength that signals both genetic quality and approachability. On the other end of the spectrum is Angelina Jolie, whose lips have achieved iconic status in modern culture. Jolie possesses what beauty editors have described as the gold standard in lip beauty. Her lips are full, symmetrical, and perfectly proportioned to her face. They balance her other features, particularly her strong jawline and high cheekbones.
The fullness of her lips signals youth, health, and high estrogen levels. They have become such a recognizable part of her appearance that they are frequently referenced in discussions of beauty standards and in cosmetic clinics where patients request procedures to achieve a similar look. The power of full lips as an attractiveness signal is perhaps most starkly demonstrated by the explosive growth of the lip augmentation industry. In the last decade alone, over 30 million lip enhancement procedures have been performed worldwide. This is an extraordinary number. It means that tens of millions of people, the vast majority of them women, have chosen to undergo medical procedures, often involving needles, injections, pain, and expense, solely to make their lips appear fuller. The most commonly requested outcome is a full, symmetrical Cupid's bow, the exact feature that naturally signals high estrogen and fertility. This trend raises interesting questions about the relationship between biology and culture. On one hand, the desire for fuller lips appears to be driven by deep biological preferences shaped by evolution. On the other hand, the specific aesthetic ideals, the exact degree of fullness considered most attractive, the shapes and proportions that are fashionable at any given moment, are clearly influenced by culture, media, and celebrity trends. The interplay between these forces is complex. One particularly troubling aspect of the lip augmentation trend is its intersection with racial and ethnic appearance. People of African descent have a genetic predisposition to fuller, more pronounced lips. This is the result of specific gene variants that are more common in populations with African ancestry, combined with other factors like melanin levels. For centuries in Western societies, these features were stigmatized, mocked, and considered unattractive. Racist stereotypes portrayed full lips as primitive or undesirable. Yet, in recent decades, as the lip augmentation industry has boomed, the very same full lips that were once derided in black individuals have become highly desirable in white individuals. Patients pay thousands of dollars to achieve through injections what many black people possess naturally. The most requested shapes and proportions in cosmetic clinics often mimic natural black features. This represents a clear case of cultural appropriation, where physical features are valued when they appear in one racial group but stigmatized when they appear in another. This history does not negate the biological basis of lip attractiveness. Evolution has shaped human preferences for certain lip characteristics because those characteristics reliably signaled fertility and health. But culture determines how those preferences are expressed, who is allowed to be considered beautiful, and which features are valued or devalued based on the race of the person displaying them. The lips themselves respond to sexual arousal in ways that reinforce their role as sexual signals. During arousal, blood flow to the genital area increases dramatically. This is well known. But blood flow also increases to other parts of the body, including the lips. The increased blood flow causes the lips to swell slightly, becoming fuller and redder. The effect is temporary, lasting only during the period of arousal, but it is noticeable. This temporary swelling makes the lips even more prominent as sexual signals, emphasizing fertility and readiness for sexual activity at precisely the moment when such signals are most relevant. The reddish color of lips, which results from blood vessels visible through the thin vermilion tissue, also functions as a fertility signal. The color indicates good circulation and oxygenation of the blood. It signals health and vitality. In light-skinned individuals, the red or pink color is quite pronounced. In darker-skinned individuals, the effect is more subtle, but the lips are still typically darker and more richly colored than the surrounding facial skin. Throughout human history, people have enhanced this natural coloring through cosmetics, from the crushed red rocks used by ancient Mesopotamians over 5,000 years ago to the modern lipsticks that dominate the beauty industry today. The use of lipstick is itself revealing. When a woman applies red or pink lipstick, she is artificially enhancing the color and definition of her lips, making them appear more youthful, fuller, and healthier than they might naturally be. She is, in effect, creating the illusion of higher estrogen levels, greater fertility, and younger age. This is not deceptive in any dishonest sense. It is simply an example of using cultural tools to amplify biological signals that already exist. Men understand, at least on some level, that lipstick is cosmetic. Yet the enhanced appearance still triggers attraction because it mimics the natural signals that evolution has wired men to respond to. The same logic applies to lip augmentation procedures. Collagen injections, hyaluronic acid fillers, and other techniques are used to restore or enhance lip volume. They create the appearance of youth and high estrogen, even in women whose natural collagen production has declined with age. The procedures are popular precisely because they work. They make women appear more attractive to potential mates by mimicking the biological features associated with fertility. This raises an interesting evolutionary question. If lips function primarily as fertility signals, why do they remain prominent throughout a woman's life, rather than disappearing after reproductive age? In many animal species, features associated with reproduction are expressed only during the fertile period and then regress. Yet human lips, while they do thin with age, do not disappear entirely. They remain visible and functional features of the face. The answer likely relates to the long human lifespan and the importance of post-reproductive individuals in human social groups. Unlike most animals, humans commonly live for decades beyond their reproductive years. Postmenopausal women play critical roles in human societies, providing care for grandchildren, transmitting cultural knowledge, and contributing to group survival. While their lips no longer function as fertility signals, they still serve other functions related to eating, speaking, and facial expression. The lips do not need to disappear simply because fertility has ended. Additionally, human mate selection is not based solely on fertility signals. While fertility is important, especially for long-term pair bonding aimed at reproduction, humans also form relationships based on many other factors, including compatibility, shared interests, emotional connection, and social status. A woman whose lips have thinned with age may no longer be at peak fertility, but she can still be attractive for reasons unrelated to reproduction. The biological costs of prominent lips must also be considered. Everted lips with exposed vermilion are vulnerable to environmental damage, drying, chapping, and injury. They are also more susceptible to certain medical conditions, including infections and cancer. Lip cancer, caused predominantly by tobacco use and sun exposure, is more common in people with prominent lips because the tissue is thin and delicate. The risk is substantial enough that it has been documented in medical literature as a public health concern. For prominent lips to have evolved despite these costs, the reproductive benefits must have outweighed the health risks. This calculation makes sense when you consider the importance of mate selection in evolutionary terms. An individual who fails to attract a high-quality mate or who fails to attract any mate at all has zero reproductive success. Their genes disappear from the population. By contrast, an individual who experiences some health problems later in life but successfully attracts a mate and produces healthy offspring has achieved the fundamental evolutionary goal. The genes that gave them attractive lips are passed to the next generation, even if those same features cause problems decades later. This is a common pattern in evolution. Many sexually selected traits carry costs. The peacock's tail is heavy and cumbersome, making the bird more vulnerable to predators. The deer's antlers require enormous energy to grow and can become tangled in vegetation. The deep voice of a human male, caused by testosterone-driven enlargement of the larynx, increases the risk of choking. Yet all these traits persist because the reproductive benefits outweigh the survival costs. Sexual selection is a powerful force, and it can favor traits that seem maladaptive from a pure survival perspective, but that provide critical advantages in the competition for mates.
In humans, the evolution of prominent, full lips represents a convergence of multiple selective pressures. Lips evolved enhanced motor control for speech, they serve functions in eating and facial expression, and they convey crucial social information through their movements and expressions. But layered on top of these functional roles is a sexual selection component. Full lips signal youth and fertility in women. Well-defined lips signal vitality and genetic quality in men. These signals are honest, difficult to fake without modern technology, and universal across human populations.
The evolution of lips cannot be fully understood without recognizing this sexual dimension. The ways in which these small structures at the center of our faces have been shaped not just by the demands of survival, but by the ancient dynamics of attraction, choice, and desire. Your lips represent a convergence of evolutionary forces spanning more than 20 million years. From the curved orbicularis oris muscle that enables precise speech articulation, to the possible waterside adaptations for consuming slippery seafood, to the ancient behavior of kissing inherited from our great ape ancestors, to their role as fertility signals shaped by sexual selection, each aspect reveals how profoundly evolution has sculpted this small but remarkable feature. What makes human lips truly extraordinary is not any single function, but rather how multiple adaptive pressures have layered meaning upon meaning onto these structures. They allow us to speak with unmatched precision, to assess potential mates through chemical signals, to bond with those we love, and to express the full range of human emotion. The next time you look in the mirror, remember, your lips are not merely features of your face. They are archives of deep evolutionary time, connecting you to ancestors who swam in ancient waters, who first learned to speak, and who passed down through millions of years this most human of traits. Thank you for listening.